talk about a program that is a very exciting program that is done jointly with NHGRI and NCI and IGMS called LINKS, that has been now implemented through a number of centers, including our centers. LINK stands for Library of Integrated Network-Based Cellular Signature. And the purpose of this program is essentially to develop a library of molecular signatures that describe how different types of cells respond to a variety of perturbing agents. And this sounds like a little bit of a crazy idea when you first look at it. It certainly looked crazy to me when I first looked at it, which was about 2 years ago, because obviously you don't really understand how sort of studying en masse how things would behave in cell lines would actually indicate how they actually potentially behave in vivo. But it turns out that when you start looking at things across a wide variety of perturbation, phenotypic assay, and cell types, you start learning very interesting things. For instance, you start learning that certain compounds may be specific to certain genetic landscapes of the cells, or that in fact there is selectivity with respect to the phenotypic assays that you're doing. This program is originally was implemented by 2 centers that were funded under the initiative at Broad Institute and HMS. In particular, they looked essentially at the genetics and the proteomics of perturbations. One thing that was done at the Broad, which was a very interesting project, was to create a reporter which is called the L1000, which is done in Luminex, where they can report for about $4 to $5 per assay on about 1,000 different, especially 1,000 genes, and these were selected to be the one most essentially orthogonal reporting on the state of the cell. So you actually even think about inferring what the actual state of the remaining 23,000 genes were based just on these 1,000 probes. And then about a year ago, several additional UO1s were added. Technology centers, including ours at Columbia, studying cellular synergy. Abroad, this is mass spec-based proteomics, metabolomics, and this is essentially almost single cell or very, very small femto level sort of antibody-based detection of proteins. And then also a set of UO1s doing computational analysis, including again one here at Columbia, Cincinnati, Miami, and the Methodist Hospital. And the quite remarkable thing about our 2 centers is that they were obviously very, very tied together and dedicated to the study of molecular synergy in different cellular contexts. So let me give you just very briefly what the aims of these projects. This is the computational center. And so first of all, we wanted to be able to develop new graph theoretical methods that allowed us to study the links profile obtained by other centers and essentially establish relationship between mechanisms that are conserved or divergent in vitro and in vivo. I'll show you some examples that are really interesting work, and I actually won't go into covering some of the work that we've done with Corey Badashen, but needless to say, we now have a very good handle on whether what we study, for instance, in a mouse or in a cell line, is actually going to be reflective of the study in vivo. We then, in aim 2, and this I'll cover a little bit more in detail, essentially developing new tools to identify cell line specific compound mechanisms of action, which we've completed, genes and drugs that can modulate drug sensitivity or resistance, and then gene and drugs that can induce specific phenotypes. And then the third aim was essentially the design of novel algorithm for the inference of gene-gene, gene-compound, and compound-compound synergy. And this, I think, was directly feeding into our second grant, which was, in fact, experimental in nature, or technology development, which essentially started with the development of appropriate high-throughput, high-content reporters to measure synergy, not just in viability, but in a variety of different phenotypes. And in fact, actually, the last cell that we're going to be screening is still open for debate, and we were hoping to maybe, out of the dialogue that emerged here, to start thinking about either motor neurons, or in fact, we have a potential phenotype with Michael Shen on an OCT4 sort of reporting cell for studying pluripotency and differentiation, lineage differentiation. And so these are, some of them have completed, and now we are finalizing the optimizations. And then in aim 2, we wanted to establish essentially the baseline. And the baseline would be, what kind of synergy would we see if we essentially started screening compounds that were as orthogonal as possible? Because one of the problems, as you see, is that many of the compounds in our current libraries do exactly the same thing. And therefore, if you start screening them just by selecting them at random, you end up heavily subsidizing a certain area of the mechanism of action as opposed to others, especially when you do that in pair. And so I'll show you what we did in terms of orthogonalizing compound mechanisms of action. And then finally, we're going to screen about 1,000 compound combinations that are now predicted to be the most synergistic according to the different phenotypes we want to report on. So let's start by predicting compound mechanisms of action. And we have two approaches that we use for compound mechanisms of action. One is based on our very well established master regulator analysis. And basically what it says is that imagine that you have a cell population, and now you have the same exact cell population but treated with a compound. So this may be actually treated just with the delivery media, say DMSO, this is DMSO plus the compound. What we would like to know is that, imagine that this is the signature or differential expression that we observed in these cells following the treatment. So these genes are actually been underexpressed, these genes are overexpressed, these genes are unchanged. What we would like to find is essentially what are the effectors of the compound activity, and we postulate that these effectors will be those that are most strikingly related to the correct regulatory mechanisms that induce the repression of these genes and activation of these genes, so something like this regulator. While regulators that in fact mostly regulate genes that have not changed will probably not be very relevant to the mechanism of action of the compound. And this is actually quite easy to understand because, for instance, what you would expect is that, let's say that we're looking for a STAT3 inhibitor, you will not see any differential expression of STAT3. But what you should see is that if STAT3 is active, then its reporter genes, that is the genes it's regulating transcriptionally, are also being expressed. But when now STAT3 is inhibited, these genes are no longer expressed. Okay? So you would like to precisely be able to monitor this effect without relying on the expression of STAT3 itself, because that would not be telling you anything. The second method is a variation of this, and now we're actually integrating the two, is an algorithmical idea which was published a few years ago by our lab. And basically it says, well, that's one metric, but the other metric is to actually measure how many of these interactions that a particular regulator is involved with change under the stress of the drug. And then we actually get more interaction or fewer interactions so that the balance may be unchanged, but in fact the activity on each one of the interactions changes dramatically. So I'll show you a couple of examples. And then the idea is essentially to map what we discover from in vivo data. So this could be either in vivo data coming, for instance, I say from ex vivo macro expression profile or protein phosphopeptide profile or whatever other type of moiety you're monitoring, where we can use that to build a regulatory model that describes the behavior of the system in an in vivo context, study mechanism of addiction or mechanism that regulate the specific phenotype of interest, and then essentially test these mechanisms by identifying potential molecules that can interact with those mechanisms at this point, not phenotypes, in vitro. And we believe that the major disconnect is that in fact, if you have a PI3 kinase inhibitor, that PI3 kinase inhibitor will actually hit the same substrate whether you do it in vitro or in vivo. The difference though is that the endpoint phenotype will be completely different because it relies not just on the fact that you're actually inhibiting the substrate, but also on all the accessory regulatory elements that may be missing, for instance, extracellular matrix signals like collagen, etc., that may be missing, or endocrine signals. And so what we do essentially here is we match what we discover in vitro from the analysis of these mechanisms of action on these compounds with what we discover in vivo, and we now have a third layer that I'm not showing here, which is where we actually perturb directly the animal. So we've done this with Cori prostate cancer, where we generate an interactome by perturbing 15 transgenic with 15 different compounds in vivo, and now we can actually relate the behavior in vivo in human, with in vivo in the mouse, with in vitro when we study the compound. So one very important thing about these analyses is that they're extraordinarily context specific. So for instance, if we're studying a breast cancer cell line, and we're studying whether this cell line will actually be addicted to a particular mechanism, and therefore there's a compound that should shut it down, we have to do that with a regulatory model that is very exquisitely specific to the breast cancer model that we're studying, and not to an arbitrary model. If we're studying a stem cell, for instance, as we're doing with Michael Shen and with Hinek Vichurle and Sarah Pribowski when we study ALS motor neuron induced from IPSs, then we need to have interactomes that are specific, for instance, to the motor neuron context, which is actually one thing that we're doing in collaboration with P2OS. So in this case, for instance, just as a proof of concept, we generated a arachne network for breast cancer using two different contexts. One was a cell line context that was generated from the CMAP. CMAP is an effort that has been performed at the Broad by Todd Golub, where he has actually perturbed with about 3,000 compounds, 3 different cellular contexts, mostly APL cell line, which is a parasitic leukemia cell line, a prostate cancer cell line, which is PC3, and a breast cancer cell line, which is MCF7. And so from all these perturbations, which have been profiled using actual Affymetrix microarray expression profile, we can actually build an interactome. We also had a very large set of TCGA-related RNA-seqs for breast cancer samples, so we generated that for in vivo context. And then we generated a integrated interactome, which now should have both the interactions that are relevant in vitro and the ones that are relevant in vivo. So there's about 1,800 transcription factors that are represented in the interactome, about 300,000 interactions. So now what we tried to do is the following. We said, can we show that these approaches are now effective at identifying mechanism of action of known compounds? So drug banks has about 220 compounds for which the mechanism of action is actually available. When overlap them with the ones in CMAP, you find about 178 compounds with 103 substrates. If you remove drugs that produce either inconsistent gene expression profiles or actually have very low RNA sequencing count, then you have about 120 compounds with 87 targets. So this is what we decided to actually test. And so the idea is that also this approach is very generalizable because we have additional data from other CMAP cell line like PC3 and HL60, and through the facility we're now generating extensive signature both in collaboration with the Broad and here at Columbia for other, and I'll show you a couple of them. So the first thing is I want to show you the negative result, which is what happens if you now try to use differential expression to study mechanism of action. So here what I'm showing is this about 78 compounds that we had, and each one of them, all the genes rank from the one most differentially expressed to the one least differentially expressed. The red bars are the actual reported substrate of that compound, so the ones that occur in GenBank, okay? Drug bank. And as you can see, essentially, there's absolutely no statistical significance. This seems to be distributed pretty much all over the place. Okay? If we now move to a virtual proteomic analysis, where now we're essentially inferring the activity of a protein from the expression of its targets, okay? Now we see a spectacular statistical significance, 10 to the minus 17, and in fact you can see that very, very frequently the substrate are right at the beginning of the and this is actually quite interesting also because we've also studied why some of the compounds may not be doing so well, and we discovered that in fact the ones that are not doing so well are the ones that correspond to proteins that have very few connections in our interactome. So if you have less than 20 connections in the interactome, you're not going to be able to predict that mechanism very well because now your reporter is not very precise or not very extensive. And the other thing that you see is that if you look at the these are the nuclear receptor classes where we have both antagonists and agonists. For the agonists it didn't work quite at all because obviously you need to be in the right regime to be able to see if that agonist is effectively working. But the p-value was, again, very, very high, very significant. You can see enrichment of all the correct substrates right at the top of the estimate of the proteins that have changed. So the negative result was in that in fact, if you take either substrate that have lower row counts from the RNA-seq, so essentially proteins that are not expressed, or actually have lower representation in the interactome, they're not going to work at all. But I think what we're saying pretty confidently now is that if our target is actually expressed, now we do, by the way, I showed you with transcription factor, but now, as we've done, for instance, with Adolfo, we can do this for signaling molecules as well with very high accuracy. In fact, our collaborations identified AKT1 as a major modulator of glucocorticoid resistance. And the idea is that through the interactome, we feel very comfortable that if the target is actually expressed in the cell, and if it's actually adequately represented in the interactome, we do a pretty good job of identifying mechanism of action of the drug. So this again generated this very nice clustering, and you can see that when you cluster by expression or by the virtual proteomic produces a much tighter clustering. And in fact, this can be used now quantitatively to assess the activity of, for instance, an estrogen receptor inhibitor, where you can see that when you use different estrogen from tamoxifen, raloxifene, fulvestrant, resveratrol, and clomiphene, you can see that essentially pretty much every time except for I think fulvestrant, which was except for Svetrol, which was used at very, very high titrations, you see dosage-specific response in the cell. Okay? And this axis shows you how much inhibition is predicted by the enrichment of the targets of the estrogen receptor according to our interactive analysis. This can also be used to identify completely novel drugs. So this is again now something for which the facility was used, where we wanted to discover. This is a long-term project of ours that we've done with Brent and Antonio Barone, and it's about discovering inhibitors of STAT3 and CP beta and CP delta, which have been shown and published to be, in fact, a major synergistic driver of the most aggressive subtype of glioma, which uses an mesenchymal signature. And so here we wanted to show whether we could actually discover, using this approach, novel compounds that could actually inhibit a particular substrate that was desired, in this case STAT3. So these are the top 3 that came out from the analysis, and toposide we also knew from an independent screen done by Brent on a large number of compounds was in fact also one of the top in terms of reducing the activity of STAT3 according to the normalized enrichment score on the interactome. What you're seeing here is that these are 3 commercially available STAT3 inhibitors, static, S3121 galactone. Static is very toxic. In fact, I think this data is probably questionable. But the other 2 show some mild suppression of STAT3 activity using a STAT3 reporter. This is essentially a STAT3 sensitive promoter fused to a luciferase compared to a vanilla reporter that was just for negative control at 2 hours, 6 hours, 24 hours. And what you can see here, 5-fluorouracil was a negative control. Etoposide was one of the positive controls, which also shows negative enrichment. And these 3 will now score. And you can see that both for monensin, for niclosamide, you now see at 24 hours there's essentially a repressive ability for STAT3 that is comparable to that of the commercially available substrate. And these were never thought to be. And now we have essentially, virtually, for instance, one thing that we're doing right now in stem cells with Michael Shen is to identify all the compounds that can either induce or revert pluripotency in compounds based on the specific regulator signature that we have inferred from interactome evolution. Okay, so now we feel reasonably good about our mechanism to identify compound mechanism of action. So how can we find now the compound to screen to test the synergy? So this is what happens if you take essentially a mechanism of action compared analysis. So red means that based on the array of mechanisms of action for all the reported regulators, these 2 compounds, the one here and here, are actually very similar. Green means that they're very dissimilar. They don't have the same master regulators. So as you can see, virtually a large portion of the compounds has essentially exactly the same mechanism of action, and you can see these very large clusters, indicating that if you were to actually select drugs at random, you would very heavily favor this cluster or this cluster or this cluster, and therefore you really would learn a lot less than the number of experiments you perform. So we decided to go and check, and here, essentially basically selecting a centroid for all the drugs, and then selecting the next drug, which has the basically most orthogonal behavior with respect to the others, and then keep adding them until you run the total number of drugs that we had. We've done the same thing, but now we've done it in independent clusters, and we actually favor this approach because it gives us better representation. The problem is that with the other approach, again, if one cluster is much, much larger than the others, the chances that you will add a compound from it, because of slight differences, will actually be increased. So this one maintains representation across the different mechanisms. And so you go essentially from this matrix, to this matrix. And so this is a matrix of the 100 compounds that are, according to our analysis, the most orthogonal among the set of compounds that we could actually have access to and that were reported in CMAP. And we've now done the analysis across the 2 other cell compounds, PC3, and in fact the HL60, and only 8 of these compounds turn out to be, in fact, as close in the other 2 columns. So we feel very comfortable now that by adding a few more contexts, we can essentially generate a universal set of compounds that should be most orthogonal and can be used for cell interrogation. So this is the initial list of compounds. Anybody who's interested can get access to this. It will also be provided on the links data set. So now for the screen, what we did is essentially took these 100 compounds and combined them with 10 compounds that were identified very similar to the modulatory profiles that Brent had done, as essentially inhibiting certain specific, very, very different mechanisms that had to do with cell viability. And so for each pair, we generated a 16-point compound combination screen using the IC50, 1/2 of IC50, 1/4, and 1/8. So this essentially starts from the dosage that is most efficiently and most accurately determined, which is the IC50, and then moves down up to 3 logs down to essentially telling us what the range of potential synergy could be. Obviously, the best synergy is when the two compounds have absolutely no activity independently and have very strong activity together. So this should explore that. Now, for any compound that is determined to be synergistic, at some of these concentration, then we go and re-evaluate with a full isobologram analysis. So this is the first matrix, and essentially what you're seeing here is in blue, we report antagonism. So this is essentially something that's called excess over bliss, and basically it is the extrapolation from what you would see from pure additivity actually done with a sigmoidal curve that has a slope of 1, which is of course a problem, so we're now also going to modify this to use the full sort of nonlinear and shape-specific response. But you get a sense of, essentially from blue, the things that should be less than additive, from red should be the things that are more than additive. And we're now looking essentially at some of these blocks where we see across multiple different time points, and this can be integrated. So for instance, here you can see that when you take omeprazole and this compound 5707A05, there's a strong synergy, and this compound also has synergy with prostaglandin A. Similar to this compound, strong synergy with omeprazole. And this is not actual synergy, but as I said, excess over bliss, which means that now we can take these specific combination and then do very accurate 12-point isobologram, 12-point by 12-point isobologram. All right, so I wanted to point out a couple of additional sets for synergy analysis that we've already done, and these have actually been used. This set is a set that was done in one cell line, LY3, which is a diffuse large B-cell lymphoma cell line, and it was the first of its kind in the sense that we did a large number of gene expression profiles, and we did them essentially using 3 replicates at 3 different time points, 6 hours, 12 hours, and 24, and 2 different concentration, IC20 and 10% of IC20. And the reason this is quite unique is that in every other experiment of this kind, experiments were done at isomolar concentration, in the sense that people put in always 1 micromolar or 10 micromolar of the compound. This is the first time where this experiment is done after very precisely titrating the compound activity in the cells, and then using that and 1/10 of that of the IC20. So dosages that were known to be sublethal for the cells, and so that they're not inducing. It's not like throwing an atomic bomb in the cell, but it's rather perturbing, hopefully, the specific mechanism of action that is associated with the compound. The other element is that this is done at 6 hours, 12 hours, 24 hours, while all the previous experiment in the CMAP are done at 6 hours only. So this gives us, and the third point is that they're done in replicates, so we have 3 data points for each one of these. And so this data was actually used for a very significant and very well-attended competition that was sponsored by the NCI called DREAM, or Dialogue on Reverse Engineering Algorithm and Assessment. And basically there were about 700 overall predictions that were submitted to this challenge. The 2 best performers were selected actually, and they have been already received their award. And Nature Biotechnology is actually going to publish the data set, essentially our algorithm, and the 2 best algorithms that have been produced by this competition. And there are similar challenges, for instance, done with Joe Gray's data on breast cancer that are now being published in the same issue in Nature Biotechnology with those results. So this was the first example where we did that, and I want to show you a couple of things. When we used the IDEA algorithm, for instance, to study mechanism of action on these compounds that were done at very high resolution, and now we have a better version of that, which Mukesh and a student in my lab have developed from original IDEA that uses far fewer data points, is much more accurate. You can see that some striking results. So this is, for instance, what happens when you perturb these LY3 cells with galvanomycin, And now we know that alpha-galvanamide is an H-shock protein inhibitor. And now what you can see in this graph, in the top 30 protein predicted by the IDEA method, is that in fact not only you have the correct substrate, but in fact many of the other H-shock proteins are in the network. And this also gives you, hopefully, this is what we expect, essentially a number of mechanisms that are downstream of the inhibited substrates, therefore characterizing what we call the network-based mechanism of action of the drug. So now the same exact network that was inferred, so we have a B-cell interactome that was inferred from a large collection of human B-cell profile, both normal and tumor-related, and that was interrogated with that signature of galvanamide. If you use the same exact network, but now with the signature obtained from cyclohexamide, you now get complete recognition of the ribosomal protein substrates, which in fact are the known substrate of cyclohexamide. And so this is a couple of examples, and we have other examples. For instance, study methotrexate and pretotrexate, which are two inhibitors of dihydrofolate reductase, which in fact cannot be at all detected as being differentially expressed. But in the two example, you get both of them. So just concluding, this is camptothecin, another example. So just showing what we're doing now with the facility. Essentially, we're extending these to 3 cell lines and the 2 FDA-approved compounds. These are the compounds, and essentially, this is just the one final thing that we also did with the facility, where we study the mTOR AKT-PI3 kinase MET axis in prostate cell lines and other cell lines, showing that there's some interesting synergy between MET inhibitors and several of the PI3 kinase AKT inhibitors that are conserved across different cell lines. So I'm going to stop here. Thank a lot of people. Obviously, this is the kind of work that the facility can do. We're very excited about being able to study synergy, not just in the context of viability, but in the context of a variety of different phenotypes, and most exciting about the ones that are stem cell related, in particular, both with our project with Dr. Sham and project with Hina Quituli and with Serge Probowski. Thanks.